Hello everyone. So today we will be discussing about insulin, and this insulin will be covered in 3 videos. So in this video my outline will be introduction, that consists of a brief history. Then we will go to the synthesis of insulin, then regulation of insulin secretion, and finally we will discuss about mechanism of action of insulin. So coming to first the introduction of insulin. So insulin was discovered by Banting and Best in the year 1921. They discovered it by demonstrating the hypoglycemic action of an extract of pancreas prepared after degeneration of the exocrine part due to the ligation of the pancreatic duct. Then extract of the pancreas. So insulin was first obtained in the pure crystalline form in 1926, and the chemical structure was fully worked out by Sanger in 1956. So now coming to the synthesis of insulin. It is synthesized by the beta cells of pancreatic isolates. And it is synthesized as a single chain polypeptide, that is proinsulin, which contains 110 amino acids. Now from this 24 amino acids are removed. Then the proinsulin is formed. The connecting or the C peptide, that is of 35 amino acids, is then split off by proteolysis in the Golgi apparatus. And the C peptide is also secreted along with insulin into the blood. Now coming to the regulation of secretion. Under the basal conditions, 1 unit of insulin is secreted per hour. So the secretion is regulated by 3 mechanisms. One is chemical, next is hormonal, and the neural mechanism. So coming to the first thing is chemical mechanism. So to understand the chemical mechanism, first we have to understand a bit of glucose homeostasis. So glucose is consumed in the food as monosaccharide or starch and disaccharide. They are metabolized to glucose. And where are they metabolized? In the intestine by an enzyme called alpha glucosidase. Okay, so the glucose absorbed in the epithelial cells of the intestine that stimulates the release of incretin. This incretin or GLP-1, this acts on the GS subtype of GPCR in the pancreas, that is the beta cells. In the beta cells, it acts on the GS subtype of the GPCR in the beta cells of the pancreas to release insulin and amylin. This amylin, it delays the gastric emptying and it induces satiety. Insulin, it acts on the GLUT4 receptors, and these receptors, they are translocated to the cell surface. Okay, this release of the insulin by the action of GLP-1, this is short, as the GLP-1, it is metabolized by DPP-4. So GLP-1 and amylin prevent the postprandial hyperglycemia. By the time the glucose reaches the systemic circulation, once the glucose reaches the systemic circulation, it stimulates the GLUT1 receptors. So GLUT1 receptors in the beta cells are stimulated once the glucose gets into systemic circulation. This glucose is taken up and metabolized to ATP. This then ATP, which is formed, what it does, it inhibits the ATP sensitive potassium channels. Okay, the ATP that is formed after the metabolism of glucose, it inhibits the ATP sensitive potassium channels. Then this inhibition of ATP sensitive potassium channel causes the partial depolarization of beta cells. This leads to influx of calcium into the cells. Then this calcium influx causes exocytic release of insulin. In normal glucose homeostasis, very little or no glucose is excreted in the urine. and this is due to the action of SGLT, sodium glucose cotransporter. SGLT that is present in the kidney. It has got two variants, SGLT1 and SGLT2. This SGLT1, it is present in the distal segment of the PCT, that is the proximal convoluted tubule. It reabsorbs or reclaims 10% of glucose that is absorbed, and the SGLT2, it is present in the early convoluted part of the proximal convoluted tubule, and it accounts for 90% of the part that is reabsorbed. So this SGLT2, it is a target for certain drugs. And these drugs are called SGLT2 inhibitors. Okay. One thing that we have to remember over here is the oral glucose is more effective in invoking insulin release than the IV glucose, and the rationale behind this is because this oral glucose generates the incretins like GLP-1, VIP and GIP. That is why it is more effective in invoking the insulin release than the IV glucose in a case of hypoglycemia. Now coming to the second mechanism of regulation of insulin secretion, that is hormonal mechanism. We all know that the alpha cells of the pancreas, they secrete glucagon, beta cells secrete insulin and amylin, and the delta cells, they secrete somatostatin. Somatostatin, it inhibits the insulin release, and glucagon, it enhances the insulin release. Now coming to the neuronal mechanism. So in the neuronal mechanism, the alpha 2 receptor stimulation, it causes inhibition of insulin release, and beta 2 receptor stimulation, it causes enhancement of insulin release. Now coming to the actions of the insulin. Actions of insulin. It decreases blood glucose by stimulating the entry of glucose into muscle and fat. by increased synthesis of GLUT4. It inhibits glycogenolysis and it also inhibits gluconeogenesis. These things are inhibited at lower concentration of insulin, and it increases glycolysis and glucogenesis. These things are aggravated at more concentration of insulin. Further, it inhibits lipolysis or breakdown of fat, and it increases the synthesis and inhibits breakdown of protein. So basically, insulin is a which hormone? Anabolic hormone or a catabolic hormone? From these things we can clearly know that insulin is a anabolic hormone. Now coming to the most important part, that is mechanism of action of insulin. So it acts on the tyrosine kinase receptors. Acts on tyrosine kinase receptors. This tyrosine kinase receptors is a heterotetrameric protein. It is a heterotetrameric glycoprotein, so to say, that consists of two extracellular alpha and two transmembrane beta, which are linked by disulfide bond. So tyrosine kinase receptors is a heterotetrameric glycoprotein which consists of two extracellular alpha subunits and two extra transmembrane beta subunits, which are linked together by disulfide bonds. The alpha subunits, they are the insulin binding sites. And the transmembrane beta subunits, they possess tyrosine protein kinase activity. So binding of the insulin to the tyrosine kinase receptors, that is the alpha subunits. For this we will see the diagram. So binding of the insulin to the alpha subunit over here, that causes aggregation and internalization of the receptors. So this receptor will be internalized. After this, what will happen? There will be phosphorylation of the insulin receptor substrate, that is IRS will be phosphorylated. Insulin receptor substrate will be phosphorylated, and after this phosphorylation, it can follow either two pathways. One is Pi3 kinase pathway, phosphoinositol triphosphate pathway, or MAP kinase pathway. So in the MAP kinase pathway, there will be transcription and translation of different proteins that will lead to the cellular proliferation and differentiation. And in the Pi3 pathway, there will be generation of the second messengers. This generation of the second messengers will cause translocation of the GLUT4 receptors to the surface, and that will facilitate more of entry of the glucose into the cell. and after this the fate of the insulin receptor is that they may be internalized and they may be degraded, or they can again be recycled to the surface. So that's all. In the next class we will be discussing about what is the preparations of insulin, the different regimens, then where it is used, its adverse effects, and after that we will discuss about diabetic ketoacidosis and hyperosmolar non-ketotic diabetic coma.